like to welcome and thank you for attending the webinar today, which is the first webinar organised by the Junior Board. I'd also like to thank our speaker, Mr. Aldo Meizoso Huesca, for agreeing to present. In case anyone's not aware, it's currently 2 a.m. in Aldo's time zone, so we're very thankful for his heroic effort today. A little bit more about Aldo. He completed an undergraduate degree in basic biomedical research at the National Autonomous University of Mexico. During his final year of the program, he completed a thesis studying different aspects of the regulation of free fatty acid receptors at the Institute of Cell Physiology in the GPCR research lab of Professor Garcia Sanz. He then continued to work at the same lab for another year as a research assistant, during which time he published a first author publication and co-authored 2 other publications. Following this, he then moved to Brisbane to commence a PhD under the supervision of Associate Professor Bradley Lanoikis, where he studies calcium-related mechanisms in skeletal muscle under different genetic and environmental conditions. Aldo is currently in his 3rd year of his PhD. His topic today is skinned muscle fibers unveil effects of the ORA inhibitor BTB in skeletal muscle. At the end of the talk, there'll be time for questions, which we'll ask you to submit through the chat as usual. Aldo, whenever you're ready, feel free to start.
Well, thanks, Silke, for such a nice introduction. Thanks to the organizers for the invitation. I'm going to start this presentation by mentioning what are our lab interests. Then I will describe a model of a study that we use in the laboratory. After this, I will describe a store-operated calcium entry, which I am sure that most of you are very aware of these mechanisms, but I'm going to describe some particularities that occur in this mechanism in skeletal muscle in specific. Once I finish this, I am going to show you some experiments that I have been doing using a store-operated calcium entry modulator, BTP2, on our particular model of a study, which here I'm already spoiling a little bit, are mechanically skinned fibers. Once I finish this section of results, I will show you an adaptation that I have been working on of the mechanically skinned fiber preparation that we think that will allow us to increase the resolution of some of the measurements that we do in the lab. After showing you this adaptation, I will show some examples of this adaptation, of these experiments, and I will finish by listing and summarizing some messages that I would like you to take home from this presentation. So let's begin. In the lab, we are interested in skeletal muscle, and we are particularly interested in a structure that is within the muscle and can be localized or observed by electron micrographies, which is this structure here known as a triad. But before talking about the triad, I just would like to go through the basic structure of a muscle fiber. For those who are not familiar with muscle, in this particular cell type, the endoplasmic reticulum is known as sarcoplasmic reticulum, and the plasma membrane of the fibers is known as sarcolemma. And talking about the sarcolemma, it turns out that this component has a very particular and remarkable feature in this particular cell type, which is that it has these deep invaginations that can be observed here. Can you see my pointer? Oh, sorry, you don't have a microphone. Sorry, I'm gonna assume that you can see it.
We can see your pointer, Aldo.
Yeah, thanks. Anyways, the sarcolemma has these deep invaginations that run from the surface to deep into the regions of the cytosol of the fiber, where it meets the structures of the sarcoplasmic reticulum. So these invaginations are known as transverse tubules or T-tubules, or as a whole, this complex invaginations is known as T-system as well. And this T-system plays a crucial role in muscle physiology as it propagates action potentials that are generated in the surface of the fiber into the cell. In the membrane of the T-tubules, these action potentials activate a voltage sensor known as dihydropyridine receptor, which is in close proximity to the ryanodine receptor in the membrane of the sarcoplasmic reticulum. The activation of the DHPR then leads to an activation of the ryanodine receptor that leads to a release of calcium into the cytosol. This calcium activates a contractile apparatus in a process known as excitation contraction coupling. So the triad is precisely this arrangement of membranes formed by a transverse tubule, shown here in black, surrounded by two structures of sarcoplasmic reticulum here and the other one here. And as we have seen in this diagram, it is very important as it establishes a platform for the interaction of several proteins that mediate different calcium-related mechanisms, which are not only excitation contraction coupling, but also store-operated calcium entry, as well as, for the sake of this talk, I'm going to mention that some passive fluxes of calcium occur within this domain. For example, SR calcium leak through the ryanodine receptor, as well as passive calcium influx through some channels that are here in the T-tubules membrane. These mechanisms are involved in different aspects of the physiology of the muscle, including force generation, adaptation, and metabolism. So overall, we are very interested in these different mechanisms that we know that occur within this triad. And in order to study these mechanisms in the lab, we use mechanically skinned fibers, which are fibers whose sarcolemma is mechanically removed with the usage of fine forceps. Main characteristic of doing this procedure is that as we peel off the membrane, the sarcolemma rolls back and forms a cuff, well, a structure that we know as cuff. And this indicates us that the fiber is actually skinned. The mechanically skinned fibers have some particularities that now I would like to mention for the purpose of this talk. The first one is that now, without the membrane, the cytosol is open to manipulation. So the cytosol in this skinned or naked fiber will be the bathing solution in which we place the fiber, which means that we can set the ionic conditions of our interest, and also we can add to this bathing solution pharmacological agents that will modulate proteins of interest for us. The second aspect that I want to mention is that in these mechanically skinned fibers, the SR calcium handling is functional. We can, for example, load the SR with a calcium-sensitive dye, in this case, fluo-5N, and we can load the SR with different cytoplasmic calcium concentrations. In these examples, 67, 28, and 200. And note how the loading of the SR calcium is proportional to the concentration of calcium in the cytosol. We can also induce depletion of SR calcium by exposing these fibers to caffeine, which activates the ryanodine receptor. And a third feature of the mechanically skinned fibers that is important for this talk to mention is that when we skin the fiber and when we peel off the membrane, the invaginations of the plasma membrane, again the T-tubules, seal over at their interface with the surface of the fiber, creating a compartmentalized extracellular space here. So this is very useful for us because this allows us to track fluxes of calcium across this membrane of the T-tubules with very high sensitivity. And the way that we do this is by isolating a single fiber under paraffin oil and exposing the fiber using a microcapillary tube to a solution, a physiological solution, containing an impermeant calcium-sensitive dye. In this case, for these experiments, we use rod-5N. So what we do basically is to expose the single fiber to the dye. When the fiber is still unskinned or intact, this dye diffuses into the lumen of the transverse tubules, the invaginations of the membrane, and we proceed to skin the fiber. As a result, the dye gets trapped inside the new compartments that we create by skinning the fiber. So this is a representative image of how the skin fiber looks, and have a look at how only the skin section of the fiber holds this calcium-sensitive dye as it gets trapped inside these structures. So in this particular model, the resting concentration of calcium inside this compartment will be the net result of fluxes of calcium that occur through the different proteins that are located in the membrane, in this membrane. And one thing that we found some years ago was actually that the ryanodine receptor calcium leak seems to contribute to the basal concentration of calcium inside this compartment. What we observe is that when we block the ryanodine receptor with either tetracaine or dantrolene, for example, we observe a decrease in the signal of Rod5N trapped inside the T tubules, corresponding to a decrease in the concentration of calcium in this compartment. Based on evidence that we have from our lab and from other groups, the current interpretation for this that we have is that the ryanodine receptor at rest is leaking calcium into this space known as the junctional space, which is the cytoplasmic space between these two membranes. This leak creates a local concentration of calcium that is higher than in the rest of the cytosol and sets the activity of PMCA that pumps calcium into the T tubules. So we can track these changes in fluorescence over time by confocal microscopy on XYT mode. And this is an example of how the trace looks when we apply tetracaine. We observe this decrease that I was talking about before. Afterwards, we can calibrate this signal by getting a maximum and a minimum value of fluorescence. And this is because we know the constant of dissociation of this dye for calcium. So having said this, now I want to use some of my time to talk about a store-operated calciumetry, which I am sure that most of you are even more experts than I am, actually, but I'll just give a brief description of it. So store-operated calciumetry, or SOC, is a mechanism of calcium influx in response to a depletion of calcium in the ER. This mechanism is orchestrated by two proteins, STIM1 in the membrane of the ER and ORA1 in the plasma membrane. And once there is a depletion of calcium inside the endoplasmic reticulum, two things happen. One is that there is an oligomerization of STIM1, and that can be observed here. And the second one is that there is a rearrangement in the membrane of the ER that brings together or brings closer the STIM1 proteins to the ORA1 proteins. The interaction between these two proteins leads to the entry of calcium as the process of store-operated calciumetry occurs. Now, in the skeletal muscle, this process also occurs, but there is a slight difference. And this is because of the intrinsic architecture of these membranes, where due to the structure of the triad, the STIM1 proteins are pre-located in close proximity to ORA1. So this has a very important consequence in skeletal muscle that can be seen in the kinetics of this mechanism in non-excitable cells compared to skeletal muscle. This is a mechanism that in the system that I just told you about, the mechanically skinned fibers, we can study. What we observe if we induce a release of calcium from the SR with the usage of caffeine, we observe that the calcium in the transverse tubules decreases almost to zero levels. So also one thing that I want to mention is that these two processes, the release from the SR and the release from the transverse tubules, can be monitored simultaneously. If we do a co-loading of dyes, Fluo5N, for example, in the SR and Rod5N in the T system, we can observe how when we expose the fibers to caffeine, we observe a decrease, an almost immediate decrease in the signal of, well, in the concentration of calcium in the SR, shown in green, that is accompanied by a decrease of calcium inside the T system, corresponding to an activation of store-operated calciumetry. But in muscle, it turns out that ORA1 not only imports calcium in response to the activation of SOC, it seems that ORA1 also imports calcium at rest into the cytosol. And the evidence that has been accumulated to that, well, that supports this idea has been done by performing some manganese-induced quenching assays. So for those who are not so familiar with this assay, I'm just going to give a brief description of it. It consists on loading cells with Fura2 in the cytosol and then exposing these cells to extracellular manganese. Now, manganese has two important properties. Well, it has way more than 2 properties, but for the purpose of this talk, there are 2 properties that I want to mention. The first one is that it gets incorporated into the cell through SOC channels. And the second one is that manganese has a higher affinity to Fura2 than calcium. So once manganese gets into the cell, it outcompetes calcium, and we observe as a consequence a quench in the signal of Fura2. This quenching can be monitored over time, and a rate of quench can be estimated. That is an indicator of the rate of entry of manganese through SOC channels. These experiments have been performed in resting myotubes in control conditions, but also in presence of BTP2, which is an inhibitor of ORI1, observing that the rate of quench in presence of BTP2 is significantly lower than in absence of this drug. Again, this is in resting myotubes, so it seems like SOC doesn't have to be fully activated in order to see this effect. The conclusion of these results is that resting myotubes, again, ORI1 channel in resting myotubes are importing calcium into the cell. So how does this idea fit in our system? Well, if this is occurring, we hypothesize that by blocking ORI1 with BTP2, we should observe if there is a flux of calcium leaking, in this case, out of the T system, we should observe an increase in calcium concentration in this compartment due to an accumulation of calcium. So I wanted to test this possibility, and in order to do that, I loaded fibers with Rod5N in their T tubules, and then I skinned them, and these fibers were exposed to increasing concentrations of of BTP2. What we observed with the first concentrations, were 1 and 5 micromolar, is what we actually expected to see, which is an increase in the concentration of calcium in response to the inhibition of ORI1. However, when we increase the concentration to 10 and 20 micromolar, we observe the opposite effect, a decline in the concentration of calcium. We thought that this could be due to 2 possibilities. One, at 10 micromolar, we are either starting to enhance a leak of calcium out of the T system, or we are impairing an uptake of calcium into the transverse tubules. Because this effect that we observed at 10 micromolar was very similar to what we observed before when blocking the ryanodine receptor, and also because there is no evidence, to the best of our knowledge, that higher concentrations of BTP2 would enhance the activity of ORI1, we decided to test whether 10 micromolar BTP2 could be affecting the ryanodine receptor. In order to do that, I repeated the same experiment. However, this time I did it in a condition where the ryanodine receptor calcium leak was constitutively blocked by a constant presence of tetracaine. So this was the experiment I performed, and well, the treatment of the first 2 concentrations, 1 and 5 micromolar, was quite comparable to what we observed before. However, the concentrations 10 and 20 micromolar, in contrast to what we observed here, where there was a decrease in the concentration, didn't exert any effect, suggesting that the decrease that we observe in this condition, that is not occurring in this condition, which doesn't have the ryanodine receptors, is due to a negative modulation of the leak of this channel here in the SR. But after looking at the leak, we decided to look on whether BTP2 10 micromolar in particular could be impairing also the release of calcium when the ryanodine receptor becomes activated. And in order to do that, I skinned fibers and I put them on a bathing solution with calcium-sensitive dye, rhodamin 2 in this case, and I looked at the amplitude of the signal when the ryanodine receptor gets activated by removing magnesium out of the solution. So this is the kind of experiments that I did. I always did a series of 2 events of calcium release. The first one would help me as a control or as a reference of a maximum amplitude of signal when the ryanodine receptor becomes activated. And the second one would be a test where I would add either vehicle or BTP2 10 micromolar. And as you can observe here, when we add vehicle, the two amplitudes are quite comparable. However, when we add BTP2, there is a reduction of about 25% on average when BTP2 is present, suggesting that this concentration of BTP2 in this particular system is impairing the SR calcium leak and calcium release through the ryanodine receptor. We then wanted to see whether BTP2 could be compromising the SR calcium uptake, or in other words, the SERCA activity. And in order to do that, I did this experiment where I loaded fibers with fluo-5N in the SR, and I performed this this exposure to increasing concentrations of cytoplasmic calcium, starting from 0 to 28, 67, and 200. Then I deplete it with caffeine and I redo it in presence of either vehicle or again BTP2 10 micromolar. As we can observe here, when we add vehicle, the second round of loading is quite comparable and quite similar to the first one. And this is the result that we obtained when adding BTP2 during the second round of loading, where we observed that calcium uptake in the SR is not only not compromised, but it is actually a little bit higher than in its own control. So far these results are suggesting that BTP2 is impairing RYR1 calcium leak and calcium release properties. However, there is a very big and important thing to consider for this experiment. And this one is that so far these experiments have been done on cytosolic administration of BTP2. Why is this important? Because so far, well, in the rest of the systems where people use intact fibers, the administration of the drug is not cytosolic, it's extracellular, to put it in some words. And so this, of course, could be affecting the results that we are observing, or could be, a reason of the results that we are observing. So in order to overcome this issue, I did the following experiment, in which, in the same way that I exposed fibers to Rod5N before skinning them, I also exposed a group of fibers to Rod5N plus 10 micromolar BTP2. Then I placed these two fibers in parallel to each other on the same chamber to be imaged. And the first thing that I did during these experiments was to see if there was an effect on cytosolic administration of BTP2 on the fibers that were pre-exposed extracellularly to the drug. So the black trace represents our control that is only exposed to Rod5N, where we can observe, as we have seen before, that the cytosolic administration of BTP2 leads to a decrease in the concentration of calcium in the T system. Now, in the fiber that was pre-exposed extracellularly to BTP2, when the fiber is exposed to cytosolic BTP2, the same drug, there is no significant effect, suggesting that the cytosolic administration of BTP2 is not exerting any effect that could not be exerted by the extracellular administration. Now, the next experiment that I did was an experiment very similar, but in this case, instead of exposing the fibers to 10 micromolar BTP2, I exposed them to tetracaine to block the leak of calcium through the ryanodine receptor. We know that when we block the ryanodine receptor in our model, we observe a decrease in the signal that is consistent here in our trace in black, which is the control that doesn't have BTP2. And in the fiber that was exposed to BTP2 extracellularly, which is the trace in red, we observe how the baseline is actually lower than the other fiber, and that once the fiber is exposed to the tetracaine, there is no significant effect. So, so far these results suggest that extracellular BTP2 belongs to some extent RYR calcium leak. But what about the release? As we have seen before, the release, the way that we study events of calcium release is by monitoring changes in the fluorescence of Rod2 when we activate the ryanodine receptor by magnesium removal. However, there is an important difference in terms of how we can handle this data. And what I want to say about this is that the signal cannot be calibrated as in these experiments. The best we can do is that we can normalize it to its own baseline. And a consequence of this is that the amplitudes that we'll observe in these experiments will always depend on a series of factors, such as, for example, just to give a couple of examples, dye quality, acquisition settings, maybe temperature, these kind of environmental conditions that can always mess up our experiments. So I was a little bit worried about this possibility, and I wanted to make sure to create, let's say, well, after my presentation, I want to hear your opinion about it, but I wanted to, let's say, have a cleaner way to do this experiment. So what I did was to try to design and implement a new modification of the mechanically skin fiber preparation. In the lab, we are calling it right now a restricted localized exposure of inhibitor in a single fiber. It basically consists on isolating a single fiber. Then we start skinning the fiber, but we leave a region of the fiber intact. And this region of the fiber intact is exposed to a solution, an extracellular solution containing two things, a calcium sensitive dye and a drug, a pharmacological agent. One thing that, and after this we finish skinning the rest of the fiber. And one thing that is essential to mention for the purpose of this experiment is that this procedure is actually performed under paraffin oil, which helps us to restrict the diffusion of the extracellular solution that we apply. So if we take this fiber into a confocal microscope and we look at it, we can see that this is how it looks, where we observe a clear distinction between a region that was exposed to the extracellular solution, and we can know where this fiber was exposed to it by the presence of the calcium sensitive dye, and the region that wasn't exposed to the dye, neither the pharmacological agent. So here I'm going to show you an example of an experiment where I did this procedure and I exposed the fiber to a solution containing fluo5N plus 50 micromolar of ryanodine, which is an inhibitor. At this concentration, it works as an inhibitor of the ryanodine receptor. And then I activated the ryanodine receptor in these fibers, inducing events of calcium waves, which are indicators of activity of the ryanodine receptor. And this is what happens. I hope you can see. Yes, I think so. So as you can see, there is a clear presence of waves when we activate the ryanodine receptors, but this propagation of waves and this occurrence of waves is only restricted to the section of the fiber that was not exposed to the extracellular solution, and we can identify it by the presence of the calcium sensitive dye. Our plan was to use then this approach to have an in preparation test and reference areas within the same fiber. And this way we wouldn't have this issue of whether the quality of one fiber compared to the other would be different, or the bathing solution was different, or things like this. So I did this experiment in which instead of exposing the fibers to ryanodine, I exposed them to BTP2. Here I did two fibers in parallel. The first one has only fluo 5N, and the other one has a fluo 5N plus BTP2. I did this in parallel in order to make sure that the sole exposure of the extracellular solution wouldn't compromise the release of calcium from the ryanodine receptor. So once I established these two fibers in the same chamber, I bathed them on a solution with cytoplasmic rhodamine 2, and I tracked the signal of rhodamine 2 on 4 different regions of interest, which are region 1 and 2, corresponding to the regions of the fiber that doesn't have BTP2, but partially has fluo 5N. Again, sorry, this is as a control to test, just to test that the extracellular exposure is not compromising the release capacity of the fiber. And the regions 3 and 4 correspond to the fiber that were exposed to BTP2. So I did these traces where I induced the opening of the ryanodine receptor by removing magnesium out of the solution, and I analyzed the amplitude of the signal when the ryanodine receptors become activated as an indicator of the activation of the ryanodine receptors again. Sorry. What we observed is that in region 1 and region 2, the amplitudes are quite comparable, suggesting that the sole application of this solution is not compromising the release capacity of the fiber. And in the regions 3 and 4, which is the third one, the one that has BTP2, we observe that there is actually a decrease in the amplitude of the signal when the ryanodine receptor becomes activated, suggesting that the presence of extracellular BTP2 that was originally exposed to the T-system lumen, which is an extracellular space, is compromising the release of calcium through the ryanodine receptors. Part of these, these experiments that I have shown you so far are part of a publication that we recently did in the Journal of General Physiology. I didn't include the whole story, so if you are keen to look at some other results and some other experiments that we performed, you're more than welcome to have a look and give some feedback if you feel like. And I will just want to finish by summarizing some messages that I would like you to take from this presentation. The first one is that mechanically skinned fibers allow the study of calcium fluxes across the T-tubules at a high sensitivity. The second one is that dissection of muscle fibers under paraffin oil allows localized exposure of pharmacological agents of interest. And in line with this, it's a third message, which is that we think that this allows us to have in preparation control and test conditions within the same fiber again. And finally, that following this procedure, we found that BTP2 negatively modulates the ryanodine receptor. So I would just like to finish by thanking the European Calcium Society for the invitation, to thank my supervisor, Bradley Leonikonis, for his support, for having me in the lab in general, to the School of Biomedical Sciences of the University of Queensland. I'm very grateful as well to them, and also to the Australian Research Council, who provided funding for the development of this research. Thanks for your attention as well, and I am happy to take any questions, comments, complaints. Please feel free. Thanks.
I might jump in here and say, first of all, thank you, Aldo, for such a great talk. That was really enlightening and really interesting. We've got a couple of questions already in the chat, and some of them are kind of directed to the specificity of BTB2, especially at that higher concentration. So there's one question, for example, from Rajesh saying that it might potentiate certain TRPM channels. Have you considered the effect that BTB2 might be having on other channels that could be in this system?
Yes, we are aware of that. We think that it might affect, for example, TRP channels. There is evidence that POMZ it could affect TRP channel 1, 3, 6, and I think there's another subtype as well. We are aware of that, and actually, because those channels are also involved in the SOC. Sorry, can I go back in the slides?
Yeah, for sure.
Yeah, because they are also involved. I mean, I think that if it was for me, I would, instead of naming here in this diagram ORI1, I would say SOC channels, because we know that BTP2 affects more than just ORI1. But in this case, for schematic purposes, let's say, I named it ORI1 as it is historically known as an ORI1 inhibitor. But yes, that is true. It can affect other channels that are present in our system.
We have a follow-on question from Gillian. What is BTB's KD and/or IC50 for ORA, and how does that compare to the concentrations at which you are seeing the ryanodine receptor effect?
Oh, I'm very sorry. I don't have the values in my mind at the moment, but I do know that they are quite lower. They are, I think they are in the nanomolar range, actually. However, we started using micromolar concentrations because, well, two reasons. Those were the concentrations that started to give us to show an effect in the system. And also that if we look in the literature of the use of BTP2 on the muscle field, it has historically been used at 5 to 10 micromolar.
Is there a difference, this is from Martin now, is there a difference in BTP2 sensitivity of coupled and uncoupled ryanodine receptor 1, PR coupled, that is?
Sorry, I didn't get the question quite well.
Yeah. Sorry, let me repeat that. Is there a difference in BTP2 sensitivity of coupled and uncoupled ryanodine receptor 1, and that's coupling to the DHPR?
Oh, okay. I am not sure, to be honest. We don't know, because we are not even sure that the effect that we observe of BTP2 on the ryanodine receptor is due to a direct modulation of this channel. We have some evidence that suggests that actually BTP2 is not acting directly on the ryanodine receptor, but somehow it is messing around with its activity.
And there's a question that sort of follows on to this, which I think you've sort of answered, which is from John Mackerel. Is there a direct effect of BTP2 on ryanodine receptor 1? Does it modify ryanodine receptor 1 in single channel experiments? Yeah, I already answered that question. This is the questions that we've got. We've got one from Giet. Have you tried any other ORI1 inhibitors?
Yeah, yes, we tried Synta 66, I think. It was a long time ago, so I think that was the name of it. But I didn't explore. No, I think the short answer for that is no. Sorry. I started trying it, but then I just focused on BTP2. That's the short answer.
And we've got a similar comment / question from Don Gill, which is that BTP is not a very specific blocker of ORA channels and has effects on TRPC channels and probably other channels. But these effects are at sub-micromolar levels. At 10 micromolar, probably many other effects. Do you have any other evidence for the action of ORA channels in triads other than BTP2?
Do I have evidence of what, sorry?
Evidence for the action of ORA channels in triads other than BTP2. So you mentioned Synta 66, but was there anything else that you've also considered?
Other drugs?
Or other ways that you've looked at the action of ORA channels?
Um, for the purpose of this project, which we initially just wanted to characterize the effect of BTB2 on the concentration of calcium in the transverse tubules, in skilled transverse tubules, I guess the answer would be no. We just wanted to focus on it and maybe potentially use it as a tool to see how much ORI1 or well, I'm gonna rephrase it, BTB2 calcium sensitive channels were leaking calcium out of the transverse tubules.
That makes sense. And is similar to what we're also seeing in the chat. So we've got a comment that at 10 micromolar, it blocks multiple classes of channels, and Kiat points out that I think we'd like to know its selective impact on ORI1 and not other channels. But Brad's also jumped in to say that 10 micromolar BTB is commonly used to block SOS in many skeletal muscle experiments, hence the interest. So it's often assumed to be specific.
Yeah, that is the main reason why we kind of wanted to see what these particular concentrations were doing in our system, because these are the concentrations that have been broadly used in order to make conclusions about the roles of SOC, for example, in skeletal muscle.
Unless we've got any more, Giat asks, are any genetic approaches feasible?
Yes, they are. There are well-known mutations of ORI1 that either blunt the activity of the channel or enhance the activity of the channel.
And is this anything that you're looking at further with this model, or?
Well, there are conditions where, for example, we know quite well patients here in Australia. I think a couple of patients that have this mutation in ORI1 that enhances the flux of calcium through these channels at rest. Okay, so what we were thinking that we could do by using something between 1 and 5 micromolar, which are the concentrations that seem to produce the effect corresponding to a inhibition on SOC channels, is to compare let's say the delta that we have from the baseline compared to when we block these these BTB2 sensitive channels. We would expect that in these individuals with a mutation that enhances the flux of calcium through ORI1, this delta would be greater. So we think that we would like to try to use this as an approach to see the contribution of ORI1 into the calcium, into the concentration of calcium in the T system, to get an idea of how much calcium at rest ORI1 is leaking. If that makes sense.
We've got a follow-on comment on that there are also mouse Nokian RYR1 malignant hypothermia mutants. And from the same questioner, John Mackerel, do you think BTB has therapeutic potential for treatment of diseases where there is a leaky SR, for example, malignant hypothermia?
Sorry, was that a question?
Yeah, sorry. Do you think BTB has therapeutic potential for treatment of diseases where there is a leaky SR, for example, in malignant hypothermia?
I don't think so. I think that it would be worth it to explore the possibility, but I think that for malignant hyperthermia, I would prefer on focusing on treatments to target like the ryanodine receptor without messing with other proteins. But yes, that's something that could be explored. And I think that before that you mentioned the question about RYR1 Nokian mice, something?
Yeah, I think this is more a general comment of other genetic approaches that could be feasible about the Nokian mice models that do exist.
Yes, we do have a Nokian model. We have both the heterozygous and the homozygous mouse model, and that is something that we were thinking on potentially exploring. There are too many things to do, so too little time to do them. So,
speaking of time, I think it's probably about time that we can open up to a more informal discussion over microphones, for example. So if anyone's got any further comments or questions that they'd like to direct at Aldo, we'll move to the more informal part of the talk. But before that, thanks again to everybody for attending the first junior